In this video, we're going to be covering the biochemical reactions that comprise glycolysis. But first, before we go into those specifics, I would like to first review the basic concept of cellular respiration, followed by having an overview of all the enzymes that looking at the classes of enzymes that participate in cellular respiration, and then we're gonna take a look at key points to focus on when we're trying to analyze each individual reaction of all these processes, most notably cellular respiration and the citric acids. Most notably glycolysis and the citric acid cycle. So with this in mind, let's get started. So first things first, cellular respiration refers to the oxidation of glucose and the reduction of oxygen to form 6CO2, which is just oxidized glucose, and 6 water molecules, which is just reduced oxygen, plus the main component and the main purpose of cellular respiration, which is to make high energy intermediates, which are comprised of potential energy, and in this case we call this this quintessential high energy intermediate ATP, or adenosine triphosphate, plus a byproduct of heat, which isn't necessarily important, but this is the key focus of what we want to investigate. So cellular respiration occurs in several places. So for and the place that cellular respiration takes place depends on the process. So for glycolysis occurs in the cytosol of eukaryotes and prokaryotes. Okay? For pyruvate processing, or the fates of pyruvate, because there's multiple pathways that pyruvate can take upon glucose being oxidized into pyruvate. So pyruvate can undergo several reactions depending on what the cell wants to produce. So in this case, pyruvate processing occurs either in the matrix of the mitochondria for eukaryotes or the cytosol of prokaryotes. And then we have the citric acid cycle, or the TCA cycle, which occurs primarily in the mitochondrial matrix for eukaryotes, because only eukaryotes have a mitochondria, right? And then lastly, the electron transport chain, or the ETC, and oxidative phosphorylation, or OXPHOS. This occurs this occurs at the inner membrane of the mitochondria, and what occurs here is an exchange of protons, hydrogen protons, right, between the intermembrane space and the mitochondrial matrix. So essentially what we have here is the IMS, the intermembrane space, and then the mitochondrial matrix. And what separates the two is the inner membrane of the mitochondria. Inner membrane. And then what contains the inner membrane space, intermembrane space, is the outer membrane of the mitochondria. Okay? We can now take a look at enzyme class nomenclature and the types of reactions occurring in cellular respiration. So the first enzyme that we're going to take a look at is oxidoreductases. So remember in the previous video of electron carriers, that what facilitates redox reactions, or the transfer of electrons, are oxidoreductases or dehydrogenases, or yes, enzymes that facilitate the transfer of electrons. The second enzyme is that second enzyme that participates in reactions in cellular respiration is transferases, or uh transferases. And what these enzymes participate in is the facilitation of a group transfer from one molecule to another, a functional group transfer from one molecule to another. And the particular type of transferase that we're interested in are kinases. And kinases are transferases that catalyze the transfer of a phosphoryl group. Okay. Hydrolases are enzymes that facilitate the hydrolysis of some sort of molecule. So, say for example, there's a molecule XY. Water reacts with the compound XY to form XOH plus YH. Okay. And in this case, we can see that the functional groups are X and Y. But it's a little more complicated than that, because what isn't necessarily transferred is whole functional groups. What essentially is transferred are whole molecules that contain carbon chains, not just individual functional groups. Okay. The next enzyme that we cover are lyases. Now, lyases, you can think of it as if it's some sort of lysis, which just refers to the cleavage of something. And in the case of lyases facilitate the cleavage of carbon-carbon, carbon-oxygen, or carbon-nitrogen bonds by elimination, leaving double bonds or rings. So in organic chemistry, what essentially happens is that there are essentially carbon-carbon bonds, right, that are formed, and by lysing them, we essentially get the formation of double bonds, right? Or the addition of groups to double bonds. So another reaction that is commonly found is that an addition that some sort of functional group would essentially react with the carbon-carbon double bond to essentially form, say for example, right, a proton transfer, which would get us some sort of functional group. Because essentially what happens here is that the double bond actually attracts electrophiles, or more specifically, or in other words, we also could possibly see the formation of a carbon ion. But essentially, that's just keep in mind that small loose detail there for now, that double bonds typically attract electrophiles, and what usually happens is the addition of these electrophiles into that, or the incorporation of these electrophiles into the overall molecule, essentially removing the double bond, as I described there. But just keep in mind for now that lyases, the key idea behind lyases, is that they essentially facilitate the cleavage of carbon-carbon, carbon-oxygen, or carbon-nitrogen bonds by elimination. Okay? And in organic chemistry, elimination just refers to the formation of a double bond by removing a bond between two atoms. Okay? The fifth enzyme that we go through are isomerases, which are just essentially enzymes that facilitate the conversion of one molecule into another, but they're essentially what we call constitutional isomers, right? Which are essentially, they have the same molecular formula, but they are essentially different in the way that the functional groups are arranged. So say for example, if we were to take a look at glucose 6-phosphate being converted into fructose 6-phosphate, you can see here that the molecular formula for both these molecules are the same, but because they are arranged differently, we would call we would consider the relationship between these two to be isomeric, right? And this transformation from glucose to fructose is achieved by an isomer raise. Okay. The sixth enzyme class that we investigate are ligases or synthetases. Okay. So ligases or synthetases are the opposite of lyases, or they're not completely opposite of lyases, but they act antagonistically towards lyases in the sense that they facilitate the formation of carbon-carbon, carbon-sulfur, carbon-oxygen, or carbon-nitrogen bonds by condensation reactions coupled to cleavage of ATP. So if I remember correctly, condensation reactions just refer to the removal of water from a molecule. And in the case of formation of carbon-carbon, in the formation of bonds, the ligases and synthetases require the cleavage of ATP. So in this case, any reaction that's facilitated by ligase or synthetase is considered an endergonic reaction because it requires ATP. It requires energy, and in this case, the energy is provided by the cleavage of ATP or ATP hydrolysis. Right? There's another form of so there's an enzyme that is similar to ligases and synthetases, which are called synthase, not synthetase. But synthase is different from synthase is almost the exact same as ligases or synthetases, in the sense that they both facilitate the formation of carbon-carbon, in the sense that they facilitate the formation of bonds, but they're different in the sense that they don't require ATP. Okay? Translocases are enzymes that facilitate the movement of molecules or ions across membranes, or the separation of molecules within membranes. So translocases, we can just consider them as transport proteins. So they just facilitate the movement of one molecule from one side of the membrane to the other. There's a couple examples shown in cellular respiration where translocases are used. Okay, so just remember that translocases are just transport proteins. And lastly, we have decarboxylase enzymes. So decarboxylase enzymes are the enzymes that catalyze non-oxidative decarboxylation reactions. So it's the removal of a carboxyl group. So it facilitates the removal of carboxyl groups. So in chemistry, we remember that carboxyl groups are usually just carboxylic acids, but in cellular respiration, we usually find carboxyl groups to be in the form of an ion. Right. So, just something to keep in mind for now. We won't see that many decarboxylase enzymes. Okay. So before we start delving into the specifics of glycolysis, I want to emphasize that there's in glycolysis that there's a huge amount of information to go over, and there's only so much that we can memorize right now, right? And in all honesty, we in this entire diagram here that shows glycolysis, I don't even consider the reaction mechanisms that take place during that characterize glycolysis, and I think that should be a separate video for another time. So for now, I want to give us a general focus on how to analyze each subsequent reaction in glycolysis. So there's several things we should consider. We want to just consider the type of reaction occurring, the class of enzyme catalyzing the reaction, so it could be any of these 9 enzymes. We want to consider the additional cofactors or coenzymes needed to stabilize high energy intermediates, and the cofactors or coenzymes that support reactions in glycolysis. So if I remember telling you guys correctly, that cofactors or coenzymes are are molecules that are non-proteins that serve as universal electron carriers. So this includes NAD, FAD, FMN, cytochromes, all these units. So NAD, FAD, or FMN, cytochromes. More specifically, the heme units. There's quinones, and then iron sulfur proteins. Okay, these are all electron carriers that, or sorry, in terms of glycolysis, I believe that the only electron carrier that we really have to be concerned about is NAD. Okay. We want another thing that we should consider when analyzing glycolysis is the high energy intermediates, because the high energy intermediates can help give us an idea of what reaction is going to occur next. So we should look for the following examples when we're looking for high energy intermediates. There's acyl phosphates. Remember that acyl phosphates are considered high energy intermediates because as they, in their currently existing form as an acyl phosphate, they have poor charge distribution, which means that an oxygen is distributed on a positive charge is distributed on the oxygen, and there's the presence of two charges in this resonance form relative to this resonance form. So there's the formation of a poor resonance hybrid in its reactant form. But if we allow the acyl group to separate from the phosphate, then we can see that the products exhibit proper resonance stabilization and better charge distribution, meaning that the negative charge here is delocalized, which allows for proper stability of the products relative to the reactants. Okay. Not to mention the existence of an inorganic phosphate group, which also contributes to the stability of the products, right? So hence this reaction is heavily favored, and thus we assume there would be a high amount of potential energy associated with acyl phosphate lysis. Okay. Thioesters, I explained this before in one of my earlier videos. I believe it was on high energy intermediate compounds. So thioesters, if I remember correctly, it also had similar reasons like acyl phosphates to exhibit or to possess a huge amount of potential energy for reactions. Okay, so if you want to take a look, just check back in this video here, energy transfers and the basics plus the role of ATP. Okay, and then we have lastly the last high energy intermediate we want to consider here are enol phosphates. So just like enols, which are just vinylic alcohols, which means an alcohol that's directly connected to a carbon that's participating in a double bond, right? Enol phosphates are literally just phosphate groups attached to a carbon that's participating in a double bond. That's essentially what an enol phosphate is. Okay. Now, along with that, there are other important notes that we need to consider about high energy intermediates. So, carbon-carbon bond lysis. There are two commonly seen carbon-carbon breaks throughout glycolysis and the citric acid cycle. Although glycolysis has a carbon-carbon bond break here. So, when a carbon-carbon break occurs, say for example, this acyl group is separated from this hydrocarbon chain, we can see that the product is resonance stabilized. That we can see that the acyl group is resonance stabilized by a prosthetic group containing a cation. in the active site of an enzyme, right? So whenever a carbon-carbon break occurs, an enzyme containing a prosthetic group will further stabilize the product and contribute to the reaction moving forward. Okay? So when a carbon ion forms, it'll be resonance stabilized, allowing this reaction to move forward. Another carbon-carbon break that we observe is the one in is any reaction involving TPP or thiamine pyrophosphate. And for this carbon-carbon break, this one is TPP mediated, since a negative charge forms on the carbon that's sp2 hybridized and that's bound to oxygen. Right? So if we see here that this pyruvate has a double bond, right? And remember when I said before that this double bond here tends to be electrophilic. It tends to be this carbon double bond oxygen bond right here, this carbon, the C double bond O bond right here, this one, it attracts electrophils. So in this case, it participates in the proton transfer with TPP. So there's a proton transfer here, and from this, a nucleophilic attack occurs from the same ring, from the same TPP ring. And then what occurs here is an arrow pushing mechanism which allows the loss of a leaving group. And in this case, we see a decarboxylation reaction occur where this carboxyl group is eliminated. Right? Because we see here that a double bond can form here, and this is in a sense an elimination reaction. But what essentially happens here is that a proton transfer actually occurs here, right? Which allows for the loss of a leaving group and a proton transfer. So there's an intramolecular reaction here for the TPP ring here, where this proton essentially donates its electrons to form a carbon to a double bond C double bond O bond right here, right? But in doing so causes the release of this molecule. And what we see here is the formation of a glyceraldehyde and the TPP molecule restored. So I don't think you necessarily need to focus too much on this right now. This isn't a very important mechanism to consider at this time. But essentially this is just one of the CC breaks that we consider. I don't think this reaction actually occurs in glycolysis, but we'll have to wait and see. With these key points in mind, I think it's safe to begin glycolysis. Actually, there is one last detail that I want to mention. So we remember that in glycolysis that there is an investment phase. There's an investment phase or preparatory phase, and then a payoff phase in glycolysis, where we see that the end result of glycolysis, we see that 4 ATP is produced and 2 NAD carriers are reduced. Okay? So in total, we have 2 ATP used in the investment. We have 2 ATP produced in total because 4 ATP is produced. 2 ATP is used in the investment phase. And we see that 2 NAD carriers are reduced. 2 NAD electron carriers are reduced, where each NAD carrier is reduced by 1 G3P molecule, right? Because if we remember from glycolysis that glucose separates into 2 G3P molecules, each reducing an NAD carrier. But we'll get into the specifics of that shortly. First things first, let's start with the basic reactions of glycolysis. So we can see here that this is just general digestion, where starch is broken down by starch amylase through lysis reactions involving water, which produces glucose and fructose. let me get a little more specific here. So uh starch reacts with water. So a hydrolysis reaction occurs where the enzyme that facilitates this reaction is starch amylase, which produces glucose. Okay. Similarly, we can also use and then we can also obtain glucose from sucrose. And remember that sucrose is just a dimer, which means a two polymer compound where, or not a dimer, it's a dipolysaccharide. It's a disaccharide, not a dimer. That's proteins. A disaccharide. Sucrose is just a disaccharide that's comprised of glucose and fructose. Right? So sucrose is broken down into glucose and fructose by facilitated by the reaction sucrase, right? And what essentially happens here is in greater production of glucose. So glucose here will essentially exist in equilibrium where it exists in its open configuration form or its closed configuration, or closed configuration. So closed configuration or cyclized even. This is its cyclized form, and this is its open form. For studying purposes, for purposes of studying glycolysis, this is the form that we'll commonly use. Glucose in its open configuration. Okay. Another molecule that's also of interest during digestion of foods would be triglycerides. And triglycerides are lipids that are essentially representative of fats, right? So again, an unknown enzyme facilitates the lysis of triglycerides to form 3 fatty acids, right? These are all fatty acids. And then glycerol. Okay. Glycerol is an important molecule that we'll need to consider later, but essentially, glycerol will be phosphorylated by ATP to form glycerol 3-phosphate, L3P, and this is to be used for complex 2B and oxphos, which is an important detail we'll come back to later. But for now, let's begin the process of glycolysis by first looking at glucose. So we see here that this is the first step. This is called Glyco 1. In Glyco 1, we see that there's a group transfer where glucose is phosphorylated by ATP to form glucose 6-phosphate. And the reaction that facilitates this is a kinase. Because we know that this enzyme is a kinase, we know that we can deduce that the reaction that occurred here was a group transfer reaction. Okay. Now, the primary purpose of glucose being phosphorylated is to keep the substrate in the cytosol through charge addition. So we don't want glucose leaking out of the. So we want to prevent glucose from leaving the membrane, from leaving the plasma membrane, right? And because the plasma membrane is commonly permeable to nonpolar substances. This would likely increase the chances, and given that glucose appears from this diagram to be an uncharged and likely unpolar molecule, although I don't believe that it's completely non-polar as a molecule, I do believe that its chances in its currently existing form as just a glucose molecule would increase its chances of leaving the membrane, right? So when we add a phosphoryl group, there's charges present, and as we know, charges present on a molecule makes the membrane impermeable to these molecules. So it's more difficult for glucose to leave. Okay, so in the second step, we call this Glyco 2. We see that glucose 6-phosphate will undergo an internal group rearrangement. So That's a very key thing that we have to understand here. An internal group rearrangement just refers to the isomerization of a molecule, where the C double bond O bond. So the C double bond O bond is this thing right here. We can see that the functional group here currently exists as an aldehyde. C double bond O is moved to carbon 2 to make a ketone. Okay? So we see here that there's an internal group rearrangement, and this is facilitated by the enzyme isomerase. Okay? This is essentially what Glyco 2 is. Isomerase facilitates the conversion of G6P to F6P. Okay? Now the purpose of converting glucose 6-phosphate to fructose 6-phosphate is to allow for upcoming phosphorylation of carbon 1 to facilitate proper resonance delocalization of the negative charge on the resulting carbon ion in Glyco 4. So just keep in mind that the key idea here is that the primary purpose right now is to phosphorylate fructose 6 to phosphorylate carbon 1 on fructose 6-phosphate to for later purposes. So we see here that fructose 6-phosphate contains a hydroxyl group on carbon 1, making it ideal for a second phosphorylation. So it'll receive the phosphoryl group by attacking the nucleus. By attacking ATP as a nucleophile. Sorry. So the nucleophile here is F6P, or fructose 6-phosphate, and it'll attack an inorganic phosphate group. And it'll attack ATP to form ADP to form fructose 1,6-bisphosphate. Okay, so there's a second phosphorylation, or a second group transfer. That's what essentially Glyco 3 is. So this is the nucleophile. It'll be attacking ATP. And ATP will undergo hydrolysis to form ADP, and we observe a second group transfer here. We also know that this is a second group transfer because the name kinase is literally in the name of the enzyme, so we know that this is a group transfer. Now the purpose of phosphorylating this compound the second time is to ensure that when fructose 1,6-bisphosphate splits, the products remain in the cytosol via charge addition. Charge addition meaning that the charges remain on on both of the molecules. Now, fructose 1,6-bisphosphate exists. Fructose 1,6-bisphosphate is going to be cleaved. And this cleavage will occur at the carbon 3 dash carbon 4 bond right here. So this big thing, this carbon bond right here, carbon 3 and carbon 4. And what happens is that we yielded 2 carbon products that are capable of interconversion. Okay. So essentially, what we have here is an aldol condensation reaction, a carbon-carbon cleavage. This is the step for Glyco 4. And the enzyme that facilitates this is a lyase, or aldolase. Okay. Now the purpose is to produce twice as much energy. So this step essentially increases the energy efficiency of glycolysis. Okay. And we see here that there are 2 products produced. Now this is a little messy. But essentially we see here that there are 2 products, G3P, or glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate, or DHAP. So there's one G3P molecule here, but as I told you before, that there's 2 G3P molecules that reduce NAD, right? So this is what the purpose of the 5th step is. The purpose is to create a second G3P molecule. So what happens here is that there will be an internal group rearrangement, and this is what the 5th enzyme serves as. So the enzyme that facilitates this reaction of DHAP into the second molecule of G3P triose phosphate isomerase, this molecule right here. Triose phosphate isomerase facilitates the conversion of DHAP to G3P. So it's an isomerase. So we see here an internal. So we see here the second group rearrangement, the second internal group rearrangement. Oh, another thing to important note is that whenever a carbon-carbon break occurs, a carbon ion will form. So because of the action of the lyase, we saw here that a carbon ion forms here, right? So it'll need to be stabilized either through resonance, prosthetic groups, or cofactors, as I described in the earlier reaction, right? So the reason why I mentioned all of this here, it will need to be stabilized either through resonance, prosthetic groups, or cofactors, is because there's an increase in the number of charges for each molecule, right? So technically the products relative to the reactants would be considered thermodynamically unstable, but because there exist prosthetic groups or cofactors, right? And the idea of resonance, the molecule becomes relatively stable compared to the reactants of the products, which is fructose 1,6-bisphosphate. And what I mean to say is that this reaction here is favorable because of several factors. Okay. Here, I'll just put this here just so that you guys can see it. Yeah, so whenever a carbon-carbon break occurs, a carbon ion will be formed. It will need to be stabilized either through resonance, prosthetic groups, or cofactors. Because the products are stabilized via resonance, the products are considered thermodynamically stable relative to the reactants, making this reaction favorable. As I said before, right? Okay. So once the second group rearrangement occurs, we have two G3P molecules. So everything. Oops. So an important note to occur that we have to consider here is that everything here occurs twice, right? Because there's two G3P molecules. So everything you see here happens twice. Okay. So G3P, or glyceraldehyde 3-phosphate, will be oxidized, as I highlight here in a different color, to reduce NAD. Okay, so there's two G3P molecules, and each G3P molecule will be oxidized to reduce NAD. Okay, and they'll be phosphorylated by an inorganic phosphate in Glyco 6. So there's no group transfer. Okay. Right? So in the case of G3P, an aldehyde, currently in its reduced form, will become a carboxylate ion because a phosphate will be placed, right? So remember, we don't want to consider this as a kinase reaction. Remember that the enzyme that facilitates the reaction for Glyco 6 is a dehydrogenase, not a kinase, right? Because that inorganic phosphate just incorporates itself into the overall molecule on its own. Okay. So we see here that NAD is reduced to NADH plus a hydrogen atom. And this occurs twice, okay? Now, the purpose of this, so the purpose of Glyco 6 is to create a high energy intermediate, and in this case, this is the acyl phosphate. So this is the acyl phosphate right here. Okay. And by creating this high energy intermediate, we can facilitate ATP synthesis. Okay? So G3P is converted into 1,3-bisphosphoglycerate. So we see here that there's 2 phosphate groups present now, not just one. Okay. So we see here that there's a phosphorylation step that occurs here for Glyco 7. So the purpose of this step is pretty straightforward. It's to synthesize ATP. So we have here a high energy intermediate, bisphosphoglycerate, and it undergoes the first ATP. It undergoes the first group transfer. This is considered substrate level phosphorylation. Substrate being that the bisphosphoglycerate phosphorylates ADP to form ATP because the reaction is favorable, right? Because, as I said before, acyl phosphate is the high energy intermediate. Okay? And what we're left with is 3-phosphoglycerate. Now, 3-phosphoglycerate will convert to 2-phosphoglycerate to prepare for the formation of a high energy intermediate in the next step. So here we have I believe the last third internal group rearrangement. So we see here that there is an isomerase, and this is called phosphoglycerate mutase, which is an isomerase. And this isomerase facilitates the conversion of 3-phosphoglycerate to 2-phosphoglycerate, where the phosphate group is the one that gets transferred to carbon 2. Right? They swap places. Okay? Now we have another no, this is not an oxidate. This is not a redox. This is not a redox reaction that's facilitated by a dehydrogenase, but I believe a redox reaction occurs here nonetheless. Okay? So what happens here is that 2-phosphoglycerate is converted into phosphophenol pyruvate, okay? But I want to first take a look at what's happening here. So essentially what we're trying to do is we're trying to form an enol, right? For Glyco 9, we're trying to for Glyco 9 or glycolysis step 9, we're trying to form an enol or a high energy intermediate to be used for the second substrate level phosphorylation. Okay. So there are 2 key parts of this reaction. One is the oxidation of carbon 2, which loses the hydrogen atom. So remember what I said before about the oxidation state of the central atom, which in this case is being carbon. I believe that this carbon atom is being oxidized because it's losing hydrogen and thus losing its electrons, right? And in the case of carbon 3, it's going to gain electrons, right? I believe this is a type of special internal redox reaction. So if you take a look here, that because C2 lost its hydrogen and forms a double bond with carbon 3, it lost its electrons, right? It lost ownership of the electrons owned by hydrogen, even if the total number of electrons is still the same for carbon number 2. But carbon 3 gains electrons, right? Because it is no longer attached to the oxygen atom, which previously had ownership of the 2 electrons that were connected to carbon, right? So this is the former relationship. This is the latter relationship. This is the former relationship for C3. This is the latter relationship for C3. Okay, that's essentially what's happening here in this whole reaction. This is an internal redox reaction, but because there's a formation of a carbon-carbon bond, because the carbon-carbon bond is formed, this is a synthase or lyase reaction. And we see here that water is lost too, right? So it's a condensation reaction. And a condensation reaction means that water is lost to form a bond between two atoms, and in this case the two atoms are carbon-carbon bonds. So just pay attention to that detail. If water is lost and CC bond forms, or a bond forms, this is a reaction facilitated by lyase or ligase. Okay. And another key detail about this, actually there's one more very important thing that we should also understand, is that there's no ATP used. No ATP used. So this can't, sorry, I made a mistake. It's not a ligase that's used, it's a synthase. No ATP, there's no ATP used. So this is so we see here that there's a condensation and condensation reaction. There's no ATP used, and there's a bond formation. Therefore, this is a synthase. Okay, this is just basic reasoning that I used here. Okay. Now, moving on to the last step of glycolysis. Okay. We see here that phosphophenol pyruvate participates in substrate-level phosphorylation, where it phosphorylates ADP to ATP. And what we're left with is pyruvate. And, well, there's two forms of pyruvate. So it directly forms pyruvate in its enol form, but it readily tautomerizes to form pyruvate. And remember that this happens twice, so everything here happens twice. So there's 2 ATP molecules produced here. There's 2 produced here. There's another 2 produced here, for a total of 4. Okay. And essentially, that's the entire process of glycolysis. Okay. All right. Now, just going a little bit further, going beyond glycolysis, we'll go through the. Actually, you know what? That is a lot to cover in a short amount of time. Or no, actually, it's not that much. There's just a little more. So essentially, what happens here is that when pyruvate forms, right, it'll have several fates that it can. There's several reaction pathways that it can take. So it can either go through the general or the common pathway where it participates in the synthesis of Acetyl-CoA, or pyruvate. It participates in the synthesis of Acetyl-CoA. It can undergo ethanol fermentation, or it can undergo lactic acid fermentation. Okay, so we'll briefly just go through these real quickly. So in lactic acid fermentation, this is just anaerobic respiration in animal tissues when lacking oxygen. So essentially what happens is that the carriers that were reduced in glycolysis, NADH, will be reoxidized to form NAD, and in the process we have we have the process, we produce lactate. So essentially, NADH is the one being oxidized, pyruvate is the one being reduced, NADH is oxidized to form NAD, pyruvate is reduced to form lactate. Okay. Now, for ethanol fermentation, a similar process is run where where pyruvate first interacts with thiamine pyrophosphate and magnesium 2 plus, that prosthetic group, right? The pyruvate dehydrogenase contains a prosthetic group, which is the magnesium 2 plus ion. And what happens is the decarboxylation of pyruvate, which forms an acetaldehyde. Acetaldehyde meaning that this is the functional group of the acetaldehyde. And keep in mind here that I still keep the numbers of the carbons as well. So essentially, the first carbon 1 participates in decarboxylation reaction, and what we're left with is carbon 2 and carbon 3, right? And essentially, this acetaldehyde participates in a redox reaction where it's reduced, and NADH is also oxidized to form ethanol. Okay? And then lastly, the synthesis of acetyl-CoA. So for the synthesis of acetyl-CoA, you can't see this sideways, but unless you turn your screen sideways, there are 5 cofactors used to convert pyruvate into acetyl-CoA. Okay? So pyruvate will interact. So I think the key thing here that we want to keep in mind here is that it just undergoes all these reactions. But the key thing here about acetyl-CoA synthesis is that while no ATP is produced, 2 electron carriers are reduced, and that's NAD and FAD. Okay. So let's take a look at the 5 cofactors very quickly. So all cofactors here are derived from vitamins or minerals, including thiamine pyrophosphate, which is derived from vitamin B1 or thiamine. Remember here that this was the this ring right here was the thiazolium ring that participated in that reaction, right? It's used in it's used in alcohol fermentation, and it's part of enzyme 1 of the PDH complex. So this is enzyme 1, pyruvate dehydrogenase. Lipoiosine is a molecule derived from lipoic acid. Okay. And these are its 2 forms, so it exists in its oxidized form or reduced form. Now, this is pantothenic acid, or the vitamin B5. So this is Acetyl-CoA is the molecule that's responsible for primarily forming acetyl-CoA, which is the thioester, right? And then we have NAD and FAD, which I already explained previously in my other videos, where they're both reduced by pyruvate. Okay? So this is essentially what we're left with. So 2 NAD carriers are reduced, 2 FAD carriers are reduced, right? And another decarboxylation reaction occurs. I'm not entirely sure where exactly it occurs, but Yes, it's the same. It's carbon one where carbon one is expelled from. So carbon one is uh is um removed from pyruvate, and what takes its and the molecule that's inserted in is the thiol-CoA, or the thioester. All right, and essentially that sets us up for the next big reaction that we're going to discuss, which is the citric acid cycle over here. Okay. Anyways, I think that's all I have to cover for now. I know this is a lot to take in, so um, if you guys have any questions, please feel free to leave a comment below. And if I made a mistake on any of this, please feel free to comment on the channel just to give me a heads up, and I'll see what I can do to fix my mistakes.